All right. People might still wander in as we go, but it is my pleasure to introduce to you our year's first official BML seminar speaker, Dr. Jamie Grimm. Jamie joins us from the Pacific Northwest, where she is a brand new assistant professor. And Jamie and I go back a little ways. Jamie did her PhD at the University of Toronto with Dr. Martin Krakosik, who also advised me for my PhD. And we shared time at a field station in remote British Columbia that is very meaningful to both of us, called Salmon Coast Field Station. So as you'll find out over the next 60 minutes or so, Jamie works very closely with Indigenous communities to generate place-based data that informs conservation and fisheries management and policy development. And she does this with her fingers in many pots and has many, many different projects that span fields like fisheries science and biodiversity governance and disease ecology, always with the goal of trying to inform our society's ability to manage biodiversity in ecologically and socially just ways. And so I'm really happy that Jamie was able to come here today and talk to our community about her work and the way that she approaches research. And so without further ado, let's welcome Jamie to BML.
Okay.
Yes.
Yeah. Okay. Hi. I am really happy to be here. Thanks so much for the invitation, and Sean for being such a gracious host over the last few days. I've already been to many more places in California than ever. I've been to Monterey Bay, I've been to Yosemite, I've been here. It's been a whirlwind and so much fun to take in these systems that are really, really new to me and really exciting. Excited to meet all of you and hopefully have some good conversation. I like to start with a bit more of an introduction to who I am and where I come from. So I am coming from mixed heritage, so Manitoba Saulteaux, which is a First Nation in southeast Manitoba, British Columbia, or Canada, if you know where that is. I also have Swiss and German heritage. I'm a first-generation university learner. I'm a daughter, a sister, and an auntie, and a knitter, beater, and climber. And these are just some of my family members, who I am. And I also like to start by acknowledging all of the project partners. As Sean mentioned, I work very closely with several Indigenous groups, which I'll mostly refer to as First Nations or nations throughout this presentation. I also work with the federal agency Fisheries and Oceans Canada, and Salmon Coast Field Station, and some other NGOs on the coast in British Columbia as well. So it's probably not surprising to anyone in this room that North America or Turtle Island is covered with a diversity of Indigenous land claims and language groups, traditional and modern territories. And that's the case also where I work, which is mostly in southern British Columbia around Vancouver Island. And there's this really heartening trend, especially in BC, where we're seeing a lot of these different First Nations groups regaining more and more of their rights to manage their own lands and waters. And that's really heartening for a number of different reasons, from the stewardship of biodiversity in these territories to just thinking about social justice in these groups regaining their rights to make decisions. But it becomes really, really complicated. As you can see, there's so many different overlapping territories here, and these are just, of course, Indigenous claims. And I like to illustrate that at the beginning of this talk to really drive home how complicated these systems can be in terms of governance. So here's a system that's representative of the areas that I often work in. We have a freshwater stream coming out and entering the ocean. And you can imagine a scenario where one First Nation might claim this territory as their own, and another First Nation might claim this other territory. And both of these nations will have had long, long histories with these territories, different priorities and methods for managing these territories and the biodiversity within. And it's not uncommon for there to be large areas of overlap in these territorial claims. Now, in Canada, most of the marine area and resources are managed by Fisheries and Oceans Canada, which is a federal governmental agency. Whereas most terrestrial and freshwater systems are actually managed by the provinces, and so in this case, British Columbia. And then when you enter industry, that will take place in one of these areas, like salmon aquaculture, which I'm going to spend about half of my talk talking about today. That industry is, of course, managed by whatever industrial organization has started it. So Grieg Seafood, for example, is a salmon farming company, and they're responsible for the management of their facilities and for the domesticated salmon populations. But being in the ocean, they need to get licenses from Fisheries and Oceans Canada, who approves whether they can operate or not. But actually, the sea floor beneath the salmon farm is leased to the industry by the province. And so you can see already that there are all these different overlapping claims to governance of these systems. And then when you consider something like wild salmon that, of course, are going to migrate through freshwater and marine systems along the coast, who has the right to manage those salmon and make decisions about what's going to happen to them? And of course, this is still a very simplified version of this problem because there's going to be many more First Nations and communities up the coast that rely and have relationships with these salmon. There's, of course, other fishing communities, both Indigenous and non-Indigenous, that rely on these salmon. There's ecology and other species that rely on these salmon. And of course, those salmon are going to interact with salmon coming from different watersheds as well. So all this leads me to the question about who has authority over decision-making. And I can't answer that question. If you can, please tell me. It's really complicated and messy at times. And this isn't just theoretical. So this is just a screenshot from a video. The structure that you can see here is a salmon farm. And if you're unfamiliar with these systems, in coastal British Columbia, salmon farms occur in the ocean environment. And the folks you see on this farm are occupying the farm in protest of its operation in their territories without their consent. So these are elders and knowledge holders, community members from the Musqueam-Danic tribal groups. And at the bottom it just says, what gives you the right to come and impact our people and our way of life? So I can't answer this question about who has authority over decision making. But what I do try to do is create a research program that takes a multidisciplinary approach that's embedded in the local context and responsive to the needs of Indigenous communities around coastal British Columbia with whom I work. And ultimately, my goal isn't to disentangle all these complicated relationships, but rather it's to provide equitable access to data that can inform decision making. And to put a finer point on it, my goal is really to redistribute data and the power that comes with it to inform sustainable fisheries. And that's why I work very closely with First Nations groups to provide this power and data to them. Okay, so when I was thinking about what I wanted to talk to you about today, as Sean mentioned, I feel like I have my fingers in many pots. I work in lots of different systems. And so I'm going to give you a breadth rather than a depth and kind of give you an overview of my research program, some things that I've done and some directions I'm going in the next little bit. And this is kind of my vision of my research program. So everything I do is rooted in this deep respect for Indigenous sovereignty and trying to uphold Indigenous rights through the ways that I do research. I often approach my research through a knowledge co-production methodology, which I'll touch on in a little bit. And then I have two main research branches, one being sustainable fisheries and one being biodiversity governance. And through this whole research process and these different research branches, my goal ultimately is to do research that's going to have tangible community impacts, both with the partner communities that I work with and for us as an academic community. Okay, so I'm going to start with just a quick definition of knowledge co-production. This is the process simply of generating research goals, methods, and outputs with research partners. This approach can help generate research that's place-based, relevant, and hopefully more trusted by end users as they're involved in the process of generating knowledge. And in that way can help bridge the gap between knowledge generation and action, either through implementation of new policy or management approaches. And it can be used, of course, to pair different types of knowledge, so things like Western science and Indigenous as well. Okay, so we'll start with some sustainable fisheries work. I also don't think I need to convince anyone in the room that Pacific salmon hold enormous value ecologically, economically, and culturally. But many populations of Pacific salmon in British Columbia are declining, and that's for a myriad of reasons, things like climate change, habitat destruction, and emerging infectious diseases, which I spend a lot of my time thinking about. And one of the routes through which emerging infectious diseases will emerge is through interactions with salmon aquaculture. So farmed salmon production has surpassed wild salmon landings in British Columbia, but also globally. So this is salmon catches. The solid line is commercially caught Pacific salmon, and that is coinciding with an increase in farmed Atlantic salmon production on the coast. And this isn't to say this is causative in any way. This is just a correlation that I'm showing you that as we're seeing declines in Pacific salmon landings, we're relying more and more on Atlantic salmon aquaculture. So if you're not familiar with salmon aquaculture, this is a little bit of an overview of what that looks like. So as I mentioned, these farms are situated in the marine environment. Most salmon aquaculture in BC is open net pen salmon aquaculture, which means below the surface here, there are these large nets that extend way down into the water column. And they have holes in them that are small enough to keep the salmon inside in most cases, but large enough to allow the free flow of water in and out of the farm. And that's really beneficial for things like temperature control, fresh water control, oxygen. It's great for the industry. But it also comes with some challenges. So a farm like this can hold up to 1 million fish in really, really tight quarters. And as we all know, that creates a really fantastic opportunity for disease to proliferate. And then the pathogens responsible for those diseases are able to flow with the seawater in and out of salmon farms, potentially causing spillover events to wild salmon populations and spillback events where wild salmon populations can infect domesticated salmon in turn. And this threat, this idea of salmon aquaculture being a reservoir that can amplify disease in the environment and threaten wild salmon, has been recognized by elders from many coastal First Nations as a key threat to wild salmon, also by the Cohen Commission, which was a federal inquiry into the declines of Fraser River sockeye, which are one of the most economically important salmon stocks on the coast, by Fisheries and Oceans Canada stock assessments, and by COSEWIC species assessments, which is an independent body that reviews the status of species to inform Canada's Species at Risk Act. So, what do we know about pathogens of salmon aquaculture? We know that pathogens are, a natural part of any healthy ecosystem. And we know that pathogens are also ubiquitous on salmon farms, so notably things like sea lice, which I'm sure you all have heard a lot about from Sean over the last couple of years, Piscine orthoreovirus, to Nasobaculum species, which are bacteria that cause things like mouth rot in Atlantic salmon. And we know that at least pathogen environmental DNA is elevated around active salmon farms versus inactive sites. And that degree of elevation can be, on average, across many pathogens, anywhere from 3 to 4 times greater likelihood of detecting pathogens at salmon farms than inactive sites. And for some pathogens, like Tenacibaculum, that odds ratio goes up to being 14 times more likely to detect this pathogen at a farm than at an inactive site. And for sea lice, we have quite a lot of data. We know that they can spread really far from salmon farms to potentially impact wild salmon populations in the same area. But we don't have any empirical data for many of these other pathogens, specifically thinking about viruses and bacteria. So that was the goal of some of my PhD research, was thinking about, well, how far can pathogen eDNA spread from salmon farms into the surrounding environment, and thus what is the potential impact or risk to wild salmon populations? So for this work, I worked with several First Nations groups, the Musqueam-Dinick Tribal Council, whose territory is up around Salmon Coast Field Station that Sean mentioned, the Mamalilakla First Nation, the Namgis First Nation, also the Colloquiet First Nation, and the Moa-Chipmutchula First Nations. And all of these orange dots are active salmon farming tenures. So these are areas where salmon farms were operating at the time of our project. Not all of these farms are in operation anymore, which I can talk more about later. Okay, so in the spirit of thinking about knowledge co-production and working with these nations, I'm going to talk you through a little bit of what that process looked like. So some of the nations that I worked with, either my PhD advisors or other collaborators had existing relationships with, and they were able to introduce me. In other cases, we didn't have existing relationships, and so it was me approaching the First Nations fresh, without pre-existing contacts. And I usually started by approaching a lot of the nations that we worked with who had a fisheries manager or a lands manager. So I'd approach that person and say, hey, like, I would like to come spend some time in your community. Here's some of the things I'm interested in. It'd be great to have some conversations and see where there might be overlaps. And so that ended up meaning spending a lot of time in this project planning process. And so that meant spending a lot of time working on projects with the nations that weren't related to my own research. So here we were capturing juvenile salmon for sea licing, which was not related to my research, or was not directly related to the research questions I was asking, but it was an area I could help in. We spent a lot of time poring over charts, thinking about where would be the best places to collect new data, and even more importantly, maybe where are the places that we shouldn't be collecting data, where are the places that are off limits to us. And with each community, I really left it up to them to determine how they would like this relationship we were developing formalized or not. And so some communities, we came up with formal research agreements that we had the university legal teams look at and their legal teams look at. And in other cases, folks were much more comfortable just relying on verbal agreements and the relationship that we had built up to that point. So we ended up going and sampling for environmental DNA across 3 regions of the coast at 9 different salmon farms. Each of the blue dots here is an eDNA sampling location, and each of the orange dots is an active salmon farm. So what we were doing was trying to spread our sampling from spots that were really, really close to the salmon farms and then extending further away up to 5 kilometers in both upstream and downstream directions. Okay, so here's some results. This plot's a lot to take in, so I'm going to walk you through it. Each of the 3 columns at the top is just a region of the coast that we did the work at, and each of the sites at the bottom was the name of a specific salmon farm that we were centering in our sampling. And on the y-axis here are a series of pathogens, salmon pathogens, that we targeted through quantitative PCR in our eDNA filters. So we worked with Fisheries and Oceans Canada to use a high throughput qPCR system where we could test every one of our samples against 48 pathogens and fish species simultaneously. And what I'm showing you here with the different colors is just the prevalence of that pathogen in our samples. And all I want you to take away from this is that there's a lot of variation. There were a couple pathogens that we didn't detect at all, and some that were super ubiquitous in the environment that were in nearly every single one of our samples, regardless of how close to the farm we were. And then, of course, some regional variation as well. And because I did this work with so many different pathogens, for the rest of this talk, we're going to focus in on one, Tenacibaculum, which, as I mentioned in the beginning, can cause diseases like mouth rot or tenacibaculosis in farmed and wild fish. And so, as you can see, this one was quite prevalent in our samples, a little bit of variation between regions. Once we had these quantitative estimates of DNA concentration for all of these different pathogens, we went through a modeling process to try to understand how far that pathogen dispersed in the environment from a salmon farm. And the way we did this was build a model that had 2 components. First was a distributed background pathogen level. Many of these pathogens are endemic in the environment, so you expect to see some background concentration arising from wild hosts that are free swimming. And then we also expected there to be a point source of pathogen DNA abundance from the farms that would decay according to some parameters as you moved further and further away from the farm. And that can differ in upstream and downstream directions. What we actually measure in the eDNA is the sum of those 2 components. And we can use mathematical modeling in this Bayesian model, or Hurdle modeling framework, to disentangle these 2 effects. So to understand the relative contributions of background and point source pathogen DNA, and also to estimate how far that DNA spreads from the point source into the environment. Okay, so I'm going to show you some results. We're going to actually start by looking at Atlantic salmon eDNA dispersal in the environment. And this is a really nice kind of proof of concept because there should be no Atlantic salmon DNA outside of the salmon farms. I say should be. So I'm going to show you a series of panels here. Each of the panels is one individual salmon farm. Each of the points is the concentration of DNA along the sampling locations, where negative values are more upstream and positive values are downstream or in the seaward direction, the direction of juvenile salmon migration. So as you can see here, point 0 is the location of the salmon farm. And as you can see, we see a peak in Atlantic salmon eDNA at the farm, as we would expect, that decays at some rate upstream and downstream. And we see that at every salmon farm we tested, unsurprisingly. Note that the location 0 changes on some of these plots, but at each point at location, for each farm at location 0, we see a peak in Atlantic salmon DNA. Oh, and here's just the fitted models. I'm not showing you the fitted models for these 3 farms just because they were really close together. So we actually had to use a different modeling framework for those 3. But I have those results that I would be happy to talk about. So again, you see a peak in Atlantic salmon DNA at the farms. Now for pathogen eDNA, the story is a lot messier, of course. The different colors and symbols here just represent different species and taxonomic groups, whether they're viruses or bacteria, for example. Again, I don't need you to notice all of these details. All I want you to take away from this is that generally at point 0, the location of the farm, For many of the pathogens, we do see a peak in DNA concentration. There's a lot more variation here, and we see that again across farms with some weird things happening in some locations. And now if we just zoom in and think about that one Tenacibaculum species that I mentioned, in some cases we're seeing obvious peaks at salmon farms, and in others, not so much. And this is just what one fitted curve to a pathogen DNA result looks like, where, again, all of the points here are the relative DNA concentration at the location of the farm. The blue is the fitted model and 95% credible intervals. And the dotted lines here are showing you the distance of spread of 95% of the expected DNA from that one pathogen. So for this pathogen, for example, again, this is Tenacibaculum at one of our farms, at the farm called Baxter. We're actually only seeing this pathogen spread a couple hundred meters away from the farm at most. In terms of thinking about this role that salmon farms play in amplifying DNA from pathogens in the environment, so this is comparing that farm source DNA versus the background DNA that we disentangled using our model. You can see that any of the yellow curves that are high above the blue, the background, these are pathogens or DNA that we're confident is elevated by the farms, versus these ones that are just ubiquitous in the environment and there's not that role of farm amplification. So we're seeing this again for several Tenacibaculum species, for Atlantic salmon DNA, of course, and also Chinook salmon DNA, which is interesting. So that is showing that these pathogens or fish species have eDNA that is more abundant or higher concentration at farms versus further away. And then again, to get back to this idea of, okay, well, how far is pathogen DNA spreading from the farms into the environment? It's really variable. I meant to take this plot out because it's hard to look at. So ignore that. Instead, what I'll just tell you is that it's variable. And for many of the pathogens we looked at, estimates are really limited by the amount of positive detections that we had. So there wasn't a lot of positive data to really constrain the model and make these estimates with confidence. But we do see the greatest median dispersal for things like Atlantic salmon DNA that we can detect in that 95% probability from the farm up to 2 or 3 kilometers away from a salmon farm. Whereas most of the pathogens like Tenacibaculum and Moritella, which is another economically important pathogen for fish, these actually only spread in the range of about 150 to 400 meters from a salmon farm, so not nearly as much as we were expecting. Okay, so now to get back to this idea of co-production. So we went into this project, we built the relationships with the nations. Some of the nations we developed these formal research agreements with. But actually, one of the nations that we did decide to develop this formal research agreement with had some changes in personnel during the duration of our project. So we signed this research agreement with the biologists who were employed by the nation. Those biologists left their positions. And then the nation's hereditary chiefs decided that they actually weren't comfortable with this research being pursued anymore in their territory because they have a strong economic relationship with the industry. And they were concerned that this research would harm that relationship or open them up to a lot of criticism from advocates who were anti-industry. And so ultimately, they decided they didn't want the data that we collected in their territory to be published, and I respected those wishes. I haven't shown you any of that data today. It didn't go into my PhD thesis. It hasn't gone into any publications. Instead, it went to into a community report that I gave to the hereditary chiefs to make their own decisions with. But it brought up a lot of questions for me that I'm still grappling with and still spend a lot of time thinking about, which is who is our research serving and who might our research be harming? Also, who has the right to approve or block research? So this particular community has both a hereditary leadership structure and an elected leadership structure. And the hereditary leaders were the ones who decided they didn't want this research pursued, while the elected chiefs did. And so it really made me think about, well, who do I follow here? Who has the right to decide? Ultimately, I decided one no was enough no's. But that also made me think about, okay, but what ethical obligations do I have to the rest of those community members and also to the public, as this was all publicly funded research? So it also doesn't feel, I don't feel totally settled with the idea that this publicly funded research also won't become public information. Okay, so ultimately, the goal of this project was to think about how far this role that farms play in amplifying pathogens in the environment can disperse into the surrounding environment. So what is the spatial extent of potential impact of salmon farms? And the hope is that we can use this kind of information for planning purposes. So for nations that, for example, are supportive of the industry operating in their territory, maybe we can use this kind of information to optimize where should we be putting salmon farms in the territory? How far does a salmon farm need to be from the migration route of a juvenile salmon to mitigate the impacts on wild salmon populations? Or how far does a salmon farm need to be placed from another salmon farm to prevent outbreaks occurring from one to the next? And the short answer is it depends a lot on tidal influence and all of these other physical factors of the environment. But it's actually quite a bit smaller than we thought it would be. I think that if we were to be conservative, placing salmon farms a few kilometers from one another would prevent outbreaks, but placing a salmon farm a few kilometers from the migration path of a wild salmon is going to be a lot more challenging. Ultimately, around the time that I was doing this work, many of the First Nations in the Broughton Archipelago, which is where Salmon Coast Field Station is, had the farms removed from their territory. So this is the same group that we saw occupying the farm in the previous slide. Through several First Nation-led processes, they were able to remove the farms from their territories, which is really incredible. And now the government, actually, I should say, I think it's the province, Sean, correct me if I'm wrong, has moved towards a consent-based model that there aren't going to be putting new salmon farms into places without First Nations consent. It doesn't always work the other way, though. Areas that salmon farms have been removed from territories where nations would like them back, that's where it gets a little messier. And I can't remember if I have a slide for this, but ultimately in Canada, the Minister of Fisheries and Oceans Canada has committed to having all salmon farms removed from BC waters by 2029. That commitment was initially supposed to happen much earlier, and it keeps getting pushed back. And there are ongoing lawsuits between the government and the industry and also First Nations. So really, the future of salmon aquaculture in British Columbia is really uncertain at this point. Okay, we'll shift gears a little bit and talk about a more concrete example of how I work in the biodiversity governance space. So I was part of a collaborative initiative that we called GOAT, Getting Our Act Together, in which we were assessing the Species at Risk Act, which is the Canadian version of the Endangered Species Act. And the way that the Species at Risk Act, or we call it SARA in Canada, the way SARA works is that there's an independent body of scientists under this umbrella called COSEWIC that I mentioned earlier, whose responsibility it is to look at all of the scientific information available and make assessments and recommendations for which species should be listed under the Species at Risk Act. And Sean serves on the board for anadromous fishes. He knows a lot more about this process than I do, so feel free to ask him. But in the early 2000s, COSEWIC and the Species at Risk Act were adapted to underscore the importance of Indigenous knowledges and Indigenous sciences in the early 2000s. So essentially, the idea was it's not enough to just look at Western science, we need to consider Indigenous science as well. So the goal of our GOAT collaboration was to review the effective inclusion of Indigenous knowledge and rights and values under the Species at Risk Act. And we did that by looking at this previously published indigenized model of conservation. So if we were to envision what does Indigenous conservation look like, what are the values that go into Indigenous conservation, these were some of the categories we were thinking about. So thinking about Indigenous stewardship of lands and waters, interconnections between species, including humans and our non-human relatives, Indigenous knowledge, place-based knowledge, and cultural values. I don't expect you to read all of this, but basically for each of these different indigenized model of conservation criteria, what we did was we scored it on how each report, sorry, I need to take a step back. We looked at all of these different status assessments that COSEWIC develops. We looked at all of them from the time COSEWIC started up until 2024, which was 800 reports. We read 800 reports and scored each one on how well it meets these different indigenized criteria. And then we came up with a composite score, which was the sum of the normalized values of each of these different criteria. So how well have we improved the inclusion of Indigenous knowledge and values in COSEWIC status assessments since the late 90s up until now? A little bit. But unfortunately, most of this increase was actually due to a new policy where COSEWIC status report authors were asked to include generic statements in the absence of Indigenous knowledge. So when they couldn't access Indigenous knowledge or any evidence of these other values for indigenized conservation, they were asked to instead include statements like, well, there's no species-specific Aboriginal traditional knowledge in this report, or we recognize that this species is important to Indigenous peoples. So most of the improvement comes from these statements being included in reports now. We also see that there's a bias, unsurprisingly, towards charismatic megafauna mammals, both terrestrial and marine, having better scores for Indigenous values than any other taxonomic group. And statistically, this is the difference. So there's a few thoughts here about what we can do or why we have done such a poor job including Indigenous knowledge and values in these status assessments. We know there's this persistent gap still between policy intent and actual practice. And ultimately, we think that having any meaningful alignment with indigenized conservation requires a governance shift, not just trying to shoehole Indigenous knowledge and values into this Western framework of status assessments. And we can see this in areas where there's more Indigenous governance. We tend to see better reports that are more aligned with Indigenous values. And as I mentioned, alignment is strongest for large, culturally significant mammals. in part because these are also large charismatic animals that have more research, more resources put towards them. A lot of these animals are also more abundant or persistent in indigenously managed landscapes. So in the northern parts of Canada, for example, where Indigenous nations have a lot more rights to management. Oh my, I only have 5 more minutes. Okay. So getting back to this idea of co-production now and thinking about community impacts. Here I was really thinking about all of the challenges that myself and many other folks have faced trying to do this really strong community-led or Indigenous-led research from within the academy. Really, really hard. So I started thinking about, okay, well, how can we shift our academic structures to make this work less hard? And as a quick introduction, this work trying to do co-production with Indigenous communities is increasing over time. So this is just the number of publications that include co-production and Indigenous over time, and we're seeing there's an exponential growth in folks trying to do this work and publishing on this work. And I think that's for a couple of reasons. I think that there's a growing recognition of the benefits of working with Indigenous communities. As I mentioned earlier, the benefits of co-producing knowledge are things like bridging that knowledge and action gap. I also argue that co-production is increasingly being seen as an ethical and sometimes legal requirement. So maybe not within our federal, Canadian, or United States legal systems, but certainly within the legal systems of the nations that we work with. But doing this kind of work comes with a lot of responsibility. This is an incomplete list, but some things are things like there's this responsibility to spend a lot of time building relationships and trust, to being accountable to our research partners, to develop truly equal partnerships, to share skills and capacity, and to co-produce all stages of the research process. So not just jumping in and taking the data you want and leaving. And fulfilling these responsibilities takes a lot of time, the understanding and respect of different knowledge systems, and the shared ownership of research and data. And I argue that our academic norms really aren't well suited to doing knowledge co-production from within the academy because of things like differing timelines and merit systems and value systems, or priorities in research questions or approaches. And I would argue actually that our conventional academic paradigms disincentivize responsible co-production because there's so much pressure for us as scientists who are evaluated by the academy to publish frequently and fast, and that is often not conducive to doing things on the timelines and respectfully with community partners. And I think that's especially true for co-production with Indigenous communities, who the academy and other Western institutions have done real violence to in the not that distant past. So again, I wanted to know, how can we do better? I'm not the first person to think about this. I'm going to just throw up a few papers, but here's a QR code that has a list of these papers if you're interested. A lot of people have been thinking about how do we do this better. But what most of the knowledge and expertise centers on that's out there is how can I, as an individual researcher, do better? How do I act when I go into community? How do I be respectful? And really what I think we need as an academic community is more support from our institutions. So what support can we get from, what challenges arise from our academic structures logistically or culturally, and what solutions can we implement? So we had a wonderful team of folks from various career stages who have experience doing this kind of work that came together. You guys will recognize Maddie, who was totally instrumentation. I can't think of the words I want. Who is very important to making this work happen. I couldn't have done it without her. We came up with a bunch of challenges and solutions through a series of workshops. We categorized them in terms of what stage in the research process the solutions should be implemented in, or the challenges arise during, or whether they're systemic in nature. And also which level of the academic hierarchy would be best suited to implement those solutions. We came up with 22 challenges. Here they are organized by how we categorized them by each individual who did the categorization, whether they thought it should be something that comes up, or it's something that comes up before you even start the project, or is more systemic in nature. I don't have time to go over all of the challenges, but to give you a taste, some of them were things like uncertainty in whether co-production is even appropriate or necessary for a given project, or things like the fact that academic institutions don't always recognize co-production and the labor that goes into it as scientific labor. In terms of solutions, we came up with 113 of them. Here we've just binned them into some categories. And we were looking at what stage of the co-production process these solutions should be implemented during. And we found that the vast majority should be implemented systemically throughout our institution, not tied to just one stage of a research project, or should be implemented before you even start a project. In terms of who might be best suited to implement the solutions, most of that onus falls to principal investigators. And I wrote this paper when I was a PhD student, but I stand by it now that I'm a PI. And again, I don't have the time to go through all 113 solutions, but I'm going to give you a couple key takeaways. So one thing we need to do is create space for Indigenous leadership in the Academy and adapt our ways of knowing and doing to be more aligned with Indigenous systems of knowledge and action, rather than expecting Indigenous folks to adapt to the Academy. We need to provide and create more long-term funding opportunities that can help build capacity and support long-term partnerships. A lot of the nations I work with can hire a biologist maybe seasonally, and so it's really hard to maintain solid personal relationships when there's so much turnover. As an academy, we should recognize that co-produced work is equally, if not more, rigorous than traditional academic research because it can incorporate all of these different types of knowledge. And finally, we need to provide flexibility for trainees. I stand by this still. I think this is something that we need to think about in terms of the coursework that students can take that count towards their degree requirements, or the timelines for them to finish their PhDs, for example. Now that I'm an assistant professor, I would maybe expand this and say flexibility for all of us and the expectations that we have for our community in the Academy. I don't think I have time to go into current projects, so I'll leave it there, but if anyone is curious, I'm happy to talk about that in the question period. Thank you.
All right, we have time for a few questions. I have a second microphone if someone wants to put up their hand for the next one too.
Great. Thanks, Jamie. So it looked like in your eDNA data that some farms had more spread of pathogens, spatial spread of pathogens than others, if I saw that correctly. Mm-hmm. And I was wondering if that's true, what factors might modulate that degree of spatial spread for different farms?
Yeah, I think over the time period we were sampling in, so each of these farms, we tried to complete the sampling transect within a day. So I think on that time scale, the thing that would modulate that the degree of spread would be mostly physical. So things like surface currents, tidal conditions, temperature in the surrounding environment. I think if we were thinking longer term, that spread would be determined by farm factors, things like the density of salmon in the farm, or things like whether there's an active outbreak occurring or not. But on this time scale, I think it would be mostly physical factors.
Thank you very much. I love that you're addressing this topic. Kia ora tato, katoa, this beautiful presentation and acknowledgement of Indigenous viewpoints. I really appreciated that you brought in the challenges and the pitfalls of doing this kind of research. I think that in itself is another exploration in itself. I also think that it's important to recognize that a lot of this research can be done internally within the tribes without the academia.
Yes, I fully agree.
And I think that recognizing that, you know, this research has been going on from the beginning, and Western science is coming in quite late in the piece, and recognizing that some of the challenges are in reparation, in decolonization, rather than you know, the tribes not responding to the academic ways of viewing things. I wonder, anyway, I'm going to stop there because I've had a lot of thoughts about this, and I've done a lot of work in that space, and I have a lot of opinions, but I'll stop there.
I would love to hear them another time.
Sure. I'd like to ask you whether any of the tribes that you work with had any ownership of aquaculture practices or revenue generation from those aquaculture activities in those regions?
Yeah, great question. None of the aquaculture operations that I worked with were nation-owned. Most aquaculture that happens on BC's coast are international companies, mostly from Norway. And in some cases, the industry does have a financial relationship with the nation, like in the case of the nation that decided they didn't want their data published. What I've heard from community members who I've spoken to is those financial benefits that come from having the industry operate in their territory are really poorly distributed within the community. So I've heard really unfortunate stories of nation leadership getting a lot of money and not pouring that money back into the community, rather seeing it as personal gain, unfortunately.
Hi, can you hear? Oh, you can hear me now. Okay, great talk. This is really cool. Forgive me because I'm gonna have to word this question while I think about it. But my question is about, first, it was really cool to see how you maybe inherited some relationships with tribes throughout your dissertation work, but also like built those relationships. And as a student myself, I'm curious, one, what was your experience like building those relationships? But also now as a PI, what are your thoughts on early career scientists, whether it's students or postdocs in very transient positions, leading those relationships? Or like, how does someone who like myself who's in this transient position, how do you like cultivate a relationship with an Indigenous partner that you know is going to be a short-term thing because your job is short-term? Does that make sense?
Yeah, that makes total sense. And I think it's a great question and something that I have been thinking about a lot as a PI. I hold my relationships really close, and I by no means would ever want to gatekeep those relationships for my students, but I also feel very cautious in who I will select to carry on those relationships as well, because this work it's really hard, and cultivating those relationships and being responsible and accountable to those relationships takes a lot of work. And so the way that I'm thinking of approaching it as I build my lab, and right now I have 2 students, both who I'm very lucky had their own relationships before joining the lab, is I think I'm going to work with students very closely to figure out what their goals are and what they want to get out of their grad student experience, and be really transparent about a lot of the challenges that come with this work. As I said, I think that consent is an ethical requirement for working on Indigenous lands and waters, but I don't think co-production is necessary for every project and for every student. And so I think I'm just going to have to be really careful about outlining the pros and cons and the challenges and benefits, and work with the student to determine if that's right for them. And then I am really lucky that now I managed to get a job in a place that I already had these wonderful relationships. So in most cases, I hope my students won't be starting from scratch the way I did. And my PhD advisors were incredible. Neither of them were as, I'm choosing my words very carefully, neither of them were like centered relationships as much as I wanted to as I was developing my research program. And again, that's fine. But so in a lot of ways, even if they had relationships, it did feel like starting from scratch and building my own relationship. And I'm hoping that I can help ease my students who want to do this type of work into these relationships and make those introductions and continue to build together. But it's really hard if you don't have an in, if you don't have someone to introduce you. I think it's still possible. I did it. It was just a lot of work. And I think you have to go in with a lot of humility and a lot of openness to asking questions and listening and knowing that you're going to make mistakes and knowing what the right approach for apologizing or making things right are when you make those mistakes. Does that answer your question at all? Does that help?
It's now 4 p.m., but not to speak for Jamie, but I'm sure Jamie would be happy answering any more questions if she wants, but I wanted to also warn people that it's 4 p.m. if you need to go. Does anyone have any more questions they want to ask? Okay, that was easy then. Thank you very much.